Hello everyone, welcome to this video on carbohydrates. This video is going to form a series that covers the content of the AQA A-level biology syllabus. This particular topic is part of the biological molecules topic, and carbohydrates are typically the first thing that are taught in the AQA biology syllabus. Today's video is specifically going to be looking at monosaccharides and disaccharides, and I will be doing a second video that covers oligosaccharides and polysaccharides. So let's talk about carbohydrates. Carbohydrates are very important in the body for providing both energy and for playing a role in structure of certain cells. So let's talk about energy. Up until now, we've probably heard about carbohydrates in the context of food, and that is because they're a very important foodstuff. So typically, a human diet should consist of around 50% carbohydrates, and these can be obtained from a number of different food sources. Things such as bread and pasta, which are considered more starchy carbohydrates, and they give longer lasting energy because the carbohydrates found within them are much larger, so they take much longer to be broken down by the body, and so they provide slow release energy. But we can also find carbohydrates in foods such as fruit and chocolate, and these often contain simpler sugars which are more easily broken down. Carbohydrates also play a role in structure, and in our next video we'll be looking at how cellulose can be used to provide rigidity to plant cells. So what are some common features of carbohydrates? Carbohydrates often form polymers, and what polymers are a bunch of monomer subunits that are all bonded together to form a larger structure. Carbohydrates all contain carbon, hydrogen, and oxygen. And carbohydrates are formed of saccharide units. So we can have monosaccharides, which are one single unit, disaccharides, that are 2 units joined together, oligosaccharides, that are anywhere between 3 and 10 units, and polysaccharides, which are multiple subunits. And the common structure for carbohydrates is CnH2nON, and N can be any number. So if we look at the carbohydrate triose, which contains 3 carbons, our formula would be C3H6O3. So let's take a look at monosaccharides. The monosaccharides that we need to cover in this course are alpha glucose, its isomer beta glucose, and what an isomer is the same atoms but arranged differently within the space, which we'll go into more in a minute. And we also need to know about fructose and galactose. Although we don't need to know these structures in detail for this course, it is important to be able to draw the skeleton structure of alpha and beta glucose, because your exam may ask questions such as how to draw a condensation reaction or a hydrolysis reaction occurring between carbohydrates, and these are typically asked using alpha or beta glucose as your subunits because they're very easy to draw. So let's take a look at that now. So to draw the skeleton structure for glucose, the simplest thing I find is to draw 2 separate structures and lay them over the top of each other. We essentially want to end up with a roughly hexagonal shape with some little sticks coming out of the side, and you'll see what I mean in a second. So the best way I find to draw this is to draw a tabletop structure and a valley structure and lay these over the top of one another. So I'll show you that now. We draw a tabletop and a valley, and you can see we've got this roughly hexagonal shape, and on the left and the right hand sides we've got 2 sticks coming out from the structure, and this now allows us to draw the atoms that are attached. So for alpha glucose, on this left hand side I've got a hydrogen atom at the top and a hydroxyl group, or an OH, at the bottom. And for alpha glucose, the right hand side mirrors the left hand side. So this is our skeleton structure for alpha glucose. Now let's draw beta glucose. So I'm going to do the same thing again. I'm going to draw a tabletop structure and a valley structure. And the beauty of these is that they don't have to be especially neat. This roughly hexagonal shape will suffice. And now we're going to label our bonds. So on this left-hand side, we're going to do the same thing again. We have a hydrogen at the top, on the top stick, a hydroxyl group on the bottom, but this time for beta glucose, the groups on the right-hand side are mirrored. So they're going to form diagonals. So on this top stick, we'll have a hydroxyl group, and on this bottom stick, we have a hydrogen atom, and you can see that they form diagonals, so that the hydrogens are diagonal to one another and the hydroxyl groups are, and it's essentially just flipped around 180 degrees. Now that we've covered monosaccharides and looked a bit more at how to draw them, let's now move on and look at disaccharides. Disaccharides are two sugar units that form when two monomer or monosaccharide subunits bond via a glycosidic bond to form a two sugar unit. So the ones that we need to know for this course are maltose, which are formed of two alpha glucose subunits, sucrose, which are formed of a fructose and a glucose subunit, and lactose, which are formed from a galactose and a glucose subunit. As I said, these disaccharides form when monosaccharides bond via a condensation reaction. So let's have a look at what a condensation reaction is and how they occur. A disaccharide forms when two monosaccharide units bond via a condensation reaction and they form a glycosidic bond. So let's have a look at how to draw this. Here we can see we've got two molecules of alpha glucose. These need to be labelled, and I've just labelled them as alpha for now, just due to spacing. So we want to draw out the substrates that are being involved in the reaction, and then we want to draw out specifically which atoms are involved in this reaction. Now, because this is a condensation reaction, we know that one of the products released is going to be water, and water's formula is H2O. Now, the reaction occurs between this hydroxyl group here on our left-hand molecule of alpha glucose and this hydroxyl group here on our right-hand molecule of alpha glucose. So when we circle the reacting atoms, we want to make sure that we circle H2O. So once we've drawn this out, I then personally like to draw an arrow and then separately draw out the products that form from this reaction. So I've moved on to a new screen here to draw the products. If I had enough room, I would draw them side by side. So I would draw my substrates for the reaction, then an arrow, and then I draw my products. So when we get two molecules of alpha glucose reacting, we get a disaccharide. And the way to draw a disaccharide is we draw two of these hexagonal glucose skeletons close to one another, and then we want to label all the atoms attached to the sticks. So we know that on these outermost sides, we have a hydrogen atom bonded to the top stick. We have a hydroxyl group on the bottom stick. Now this is where it gets a bit more tricky. So on these top sticks, on the innermost side, we still have our hydrogen atoms like normal. However, these two monosaccharides have now bonded, and the bond between them is joined by this oxygen atom. And this is known as a glycosidic bond, and it's very important that we label this to show what sort of bond it is. When we go on to later videos talking about lipids and proteins, those molecules can also bond via a condensation reaction, but they have a different type of bond. Now it's very important that we label all of our products, and as we said, because it's a condensation reaction, we know that we've also lost a molecule of water. So we can put plus H2O, or you can do an arrow coming away from the glycosidic bond to show that water has been released. And the final thing that we need to do is to label our disaccharide. So we know that our subunits were alpha glucose, and 2 alpha glucoses make the disaccharide maltose. So as well as using monosaccharides to form disaccharides, we can also then split disaccharides back up into monosaccharides. And for this, we use a hydrolysis reaction. And this is the reverse of a condensation reaction. And what hydrolysis means, if we break down our word, we've got hydro, which means water, and lysis, which means to split. So hydrolysis means to split with water. Now you can see down here, I've got our maltose subunit. That's our disaccharide made from 2 alpha glucose subunits. And I have a molecule of water. Now you can see here, I've drawn an arrow pointing to our glycosidic bond. And that's because we are using water to split up our glycosidic bond. And when this happens, excuse me, this glycosidic bond splits, and instead of maltose, we end up with our 2 alpha glucose subunits. Those hydroxyl groups are a bit poorly drawn. I underestimated the amount of space I would need, but yes. So when we draw out a hydrolysis reaction, like with a condensation reaction, it's very important that we label the products that are involved in our reaction. So in this case, it's our disaccharide, or more specifically maltose for this particular example. We need to draw our water molecule with the arrow pointing to the glycosidic bond. And then we need to draw out the product of the reaction, which in this case were 2 alpha glucose subunits. When we talk about sugars, we can not only class them as monosaccharides, disaccharides, polysaccharides, etc., but we can also class them as reducing and non-reducing sugars. So the reducing sugars that we've looked at so far for the monosaccharides are glucose, galactose and fructose, and for the disaccharides are maltose and lactose. But what about sucrose? Well, sucrose is classed as a non-reducing sugar, and we can test for the differences between reducing and non-reducing sugars in a solution by performing the following tests. So to determine if a reducing sugar is present, we first take a sample of our test substance and we add Benedict's reagent. And this test tube is then put into a water bath that's been brought to the boil for about 5 minutes, and this will allow a coloured precipitate to form. The precipitate colours range from blue if there's no reducing sugar present, and then transform to green, yellow, orange, or brick red, and the strength of the solution will determine the colour of the precipitate formed. So if there's a small amount of reducing sugar present within the substance that's being tested, then the precipitate colour may appear as green. However, if there's a lot of reducing sugar present, it may give a brick red precipitate. Now, to get a quantitative measure of the amount of precipitate or the amount of reducing sugar present, we can filter the solution and weigh the precipitate that is left on the filter paper. However, if we perform this test and a blue precipitate is obtained, it doesn't rule out that there could be a non-reducing sugar present in the solution. So to test to see if there is a non-reducing sugar present, we perform the following test. We take a new sample of our test solution, and this time we add hydrochloric acid, and it is then put into the water bath for around 5 minutes, and this needs to be a water bath that's been brought to the boil. And what the hydrochloric acid will do is it will break down any sugars present into their monosaccharide subunits. So after 5 minutes, the test tube is removed from the water bath, and we neutralise the solution with sodium hydrogen carbonate so that it's back to a more neutral pH rather than being very acidic. And then finally we add Benedict reagent, and we repeat this test so it goes back into the water bath and we see the colour of the precipitate that forms. So if the precipitate is blue, it means that there wasn't a non-reducing sugar present within the solution, and that perhaps maybe the solution is made up with other biological molecules such as proteins or lipids. Or if it gives a brick red precipitate, it means that there is a non-reducing sugar present. So let's recap everything that we've covered today. Carbohydrates have an important role in energy provision and structure. They can be classed as mono, di, oligo, and polysaccharides. They bond via a condensation reaction, which releases a molecule of water, and they can be split via a hydrolysis reaction. And a Benedict's test is used to test for reducing sugar. So now that we've covered this topic, it's important to go back and recap the material to ensure that you fully understand it. Once you think you do fully understand it, test yourself with the following test questions. Name 3 types of monosaccharide and 3 types of disaccharide. Draw the skeleton structures for alpha and beta glucose. Draw the substrates, product and byproduct needed to make maltose. Describe how you'd perform a Benedict's test for reducing sugars. What extra steps are required to test for a non-reducing sugar? That's all for today. Thank you for listening.